Welcome to The Explainer. Today, we're dissecting the absolute mechanical genius of the CRISPR-Cas9 enzyme. We're going to take all that abstract biology and turn it into a tangible working blueprint. Why? Because this is the exact tool that's revolutionizing medicine right now and, honestly, paving the way to cure genetic diseases. It's a game changer, and we're going under the hood.
To really grasp the massive scale of what we're talking about here, imagine trying to find and edit a single specific sentence hidden away in a 3 billion letter book. Now imagine you could just cut it out, fix a tiny typo, or replace it with something entirely. That's essentially what CRISPR-Cas9 does. But, instead of a printed book, it's doing this on the DNA right inside living cells. Mind-blowing, right?
So, here's our blueprint for today. We're going to meet the Cas9 machine itself, check out the guide RNA GPS, look at the PAM landing signal, walk through the step-by-step cut, and finally, look at some engineered upgrades. Okay, let's dive right in. Part 1, Meet the Cas9 machine, specifically looking at SpCas9's bilobed architecture. We're starting with the actual hardware here, nature's most programmable set of molecular scissors. So, SpCas9 is this massive protein. It's made up of exactly 1,368 amino acids. And if you look at its shape, it honestly looks kind of like a giant crab claw, just waiting to be activated. On its own, it basically just wanders around in this partially open, totally inactive state. It literally won't become an active nuclease until it gets exactly the right signal. And this shows off the brilliant division of labor inside that crab claw structure. You've got the upper RRC, or recognition lobe, which acts as the, recognition center. It binds and holds the RNA-DNA hybrid firmly in place. Meanwhile, down below, the NUC, or nuclease lobe, holds all the catalytic machinery that actually slices the DNA. It's a perfect separation of identification and execution. Find the target, then make the cut. Moving on to Part 2, the guide RNA GPS, programming the target. So if the protein is the hardware, let's talk about the software powering this machine. That's the guide RNA. It acts just like a highly specific GPS system, telling the enzyme the exact address to go to and cut. You change the guide RNA, you completely change the destination. Now, it is absolutely fascinating how scientists took nature's super complex two-part system and just simplified it. See, in the natural bacterial immune system, this whole process actually requires two totally separate molecules working together, the target CRISPR RNA and the scaffold track RNA. So figuring out how to fuse those together into one single guide RNA, that was a massive, massive breakthrough for biotechnology. This synthetic fusion, the single guide RNA, or sgRNA, is brilliantly simple. It has this conserved scaffold region at the back that you never even have to redesign. It's just there to recruit the Cas9 enzyme, like a handle. But right at the front, it has a programmable 20-nucleotide spacer. And here's the crazy part. By swapping out just that little spacer on the sgRNA, you can reprogram the entire Cas9 machine to target virtually any genetic sequence. And incredibly, you can do this for about $10 just by ordering your custom sequence online. Seriously, $10 to reprogram DNA. But actually, scratch that. Not all 20 of those nucleosides carry the same weight. The absolute crucial point for accuracy is what we call the seed region. This is a highly sensitive zone made up of the roughly 12 nucleotides closest to the PAM sequence. Even a tiny single mismatch right here acts as a strict failsafe to prevent the enzyme from cutting the wrong genomic address. So if you're out there designing a target, the uniqueness of this specific seed region is what you care about the absolute most. All right, Part 3. The PAM landing signal, Cas9's checkpoint. Because before any actual cutting happens, the enzyme has to pass a very strict security checkpoint called the PAM.
Now you might be sitting there wondering, wait, if the guide RNA is already this perfect GPS with a highly specific seed region, why in the world does the Cas9 machine even need this extra PAM sequence to function? What's the point?
Well, the PAM is fundamentally an ancient bacterial safeguard. Honestly, without it, the bacteria would accidentally destroy their own stored immune memory. You see, bacteria store little fragments of viral DNA, but they store them without the PAM attached. So, it's the ultimate self versus non-self recognition test. And for the Cas9 enzyme itself, physically binding to this PAM sequence using a specific contact domain is the actual trigger that causes the local DNA strands to unwind. For SpCas9 specifically, it's always hunting for an NGG sequence. If it doesn't find it, it won't even attempt to open the DNA. It just moves on. And let's clear up a massive, really common point of confusion right now. The PAM sequence exists only on the target genomic DNA. It is completely separate from your guide RNA. It's basically a fixed sequence sitting right next to your target site in the genome. It is absolutely not something carried by the RNA itself.
Okay, part 4, the step-by-step cut, executing the cleavage. Now we finally get to the action. The machine's assembled, the GPS is locked in, the landing signal is cleared. So how does this machine actually execute the cut once the target is found?
You know, it's not actually instantaneous. It's this rapid, highly dynamic 3-beat mechanical process. First, it scans the genome for a PAM, literally bouncing and sliding along the DNA strand. Then it melts the DNA open to form what's called a 3-stranded R loop. And finally, boom, it snaps both strands using those catalytic domains we talked about earlier.
When those two nucleus domains down in the nuc lobes strike, they sever the DNA exactly 3 base pairs upstream of the PAM. This gives you a really clean, blunt-ended double-strand break. And once this break is made, Cas9's job is pretty much done. The cell's own natural repair mechanisms have to step in to fix the break. And that repair process, that is exactly where genetic edits are permanently made.
Finally, part 5, engineered upgrades, hacking the machine. To wrap up our whole anatomical tour today, let's look at how scientists have upgraded this natural wild-type machine into a whole suite of specialized nanotechnology. Because, guess what? We're no longer limited to just what nature provided. By going in and mutating very specific amino acids, researchers have built some crazy stuff. They've made dead Cas9s that only bind to DNA without cutting it at all. They've found smaller variants from different bacteria that are way easier to deliver into human cells, and they've even engineered versions like SpI-RI that barely need a PAM at all. That practically unlocks the entire genome for targeting. It's a total game changer.
One incredibly brilliant trick for advanced editing is using these nick face variants. Basically, by disabling just one cutting domain, you can make a super precise single-strand nick instead of completely breaking the DNA in half. Like, if you mutate the D10 position, it inactivates the RuVC domain, so you only nick the target strand. But if you mutate H840, it inactivates the HNH domain, nicking the non-target strand. This level of precision is exactly what powers advanced, much safer techniques like base editing, where a full double-strand break would just be way too destructive to the cell.
So with all these mechanical upgrades basically stripping away almost all the original biological targeting constraints, it leaves us with a pretty profound question. What limits actually remain on our ability to rewrite the code of life? Seriously, if we can just engineer the hardware to target literally anywhere and cut with this kind of molecular precision, the future of genetic editing might just be bound by our imagination. Thanks so much for joining me on this explainer, and as always, keep questioning what's possible.